In today's lesson, I'd like to briefly run through some of the key differences in protein synthesis between eukaryotes and prokaryotes, and then dive into operons, which are found in prokaryotes. All right, etymology. Anytime you see this word, it just means studying the root origin of the word. It's what we've been doing every single time I've been trying to give you the pieces of Greek or Latin words that we're talking about. So when we're talking about a prokaryote, it's actually an evolutionary term. It's assuming that prokaryotes are simpler and perhaps they were the first organisms. I think it's all not very well substantiated, but it's lasted and stayed in with the name. So you've got pro, meaning before, the first, whatever, and then karyo is for the nucleus. EU can sometimes mean true or thereabouts, and so this is talking about no nucleus, and this is talking about true nucleus, or has a nucleus, okay? That's a big difference. That means that you need to memorize this right here. The main difference between prokaryotes and eukaryotes are the presence of these membrane-bound organelles. Well, what are those? Well, that's the nucleus. That has its own membrane. Mitochondria has its own set of membranes, chloroplasts, etc. All these massive structures that you have as a eukaryote cell, the bacteria do not. So all prokaryotes are bacteria, all bacteria are prokaryotes. Every other kingdom belongs to a eukaryote. So everybody else has fancier cells than the bacteria. That doesn't make bacteria simple. It just means bacteria don't have the same machinery that the rest of life does. Okay. The ribosomes, just basic idea. How does the ribosome know where to attach? Well, they've got a section that's very rich in purines, the A's and G section. They call it the Shine-Dalgarno sequence after the researchers that discovered it. And that's where it knows to clamp on. Keep in mind that you've got with the prokaryotes, you have a circular piece of DNA. You don't have, it's hard to recognize a starting point in a circle. So when they have these rich in A and G section, that's where the ribosome knows, okay, that's where I have to start. So when the DNA starts to make the mRNA transcript starts to come off the back, as the ribosomes will clamp on and begin to read. If you can imagine there being the little piece of mRNA coming off here, the ribosomes will clamp on and begin to read, and as soon as they hit that Shine-Dalgarno sequence, that's where they know how to attach. Okay. Ours has, remember we've got the post-transcriptional modifications. We put on a 5' cap with the 7-methylguanosine. So our ribosomes know that this is the start point for our mRNA with the 7-methylguanosine, with the 5' cap. Okay, big difference. Bacteria don't really go through mitosis. They go through a different process called binary fission. And that's basically just double up the DNA, grow in size, split down the middle. Now I've got 2 bacteria. Okay? And then, of course, we go through the fancy process of mitosis, which we already covered in grade 10 and then reviewed a little bit in grade 11. Size, well, these guys are very, very, very small. Single-celled, obviously, and whereas eukaryotes are mostly multicellular. There's very, very few single-celled eukaryotes, although yeast would be an example of a single cell. And so we're generally much, much, much larger because we have all that fancy machinery. We've got to have a place to put it. And so we're generally way larger, anywhere from 10 to 100 times larger than your typical bacteria cell. These guys are less than 2 micrometers in diameter. That's 2 millionths of a meter. But they're still 2 micrometers, which means I can see them with a microscope. Okay, you can see them underneath the microscope. So again, they've got 1 circular chromosome. They also have a bunch of little plasmids, but they're not considered the main chromosome. We'll talk about that in a different lesson. And of course, eukaryotes have many linear chromosomes. Humans have 46 of them, 23 pairs. 23 came from your mother, 23 came from your father. Other organisms that are eukaryotes would have a different number. So 46 would be what humans have. If you look at a chimpanzee, they got 48 or something like that. There are some plants that have over 250 chromosomes. So it just depends on what your species is, however many you're going to have, right? Protein synthesis is a little different as well. In the prokaryote, they don't have a nucleus. So for us, the transcription has to happen in the nucleus. That's where we make our mRNA. Then that moves out of the nucleus into the cytoplasm. Ribosomes clamp on, and next thing you know, I'm translating it into a polypeptide chain. Well, those are 2 very different locations with a massive wall in between with the nucleus membrane, whereas these guys don't have a nucleus to worry about. So when their transcription starts and the mRNA starts to be produced, then the ribosomes can clamp on right away, and they can start making that protein right away. So that's called coupled transcription translation. If you're interested, there's more information in the old text section. How do they know where the start codon is? Well, these guys do a little bit of extra work. They make sure that they tag the methionine with a formal group. So in other words, after you've made the protein, that methionine has already been tagged when it got added in, and that's how you know where the beginning of that protein is. Whereas we don't have anything like that. We don't need to worry about that. As far as introns go, remember we said that introns are, we'll go over here to eukaryotes, introns are present, but they have to get removed during post-transcriptional modifications. These are sections within the gene that don't actually code for the final protein product, so they get spliced out by spliceosomes. Well, bacteria don't have those. That means that their entire gene is going to code for the protein. Okay? And then operons will be our next conversation. And yes, they have them. Mostly no, although we're discovering some operons in eukaryotic genome as well. So that's kind of an exciting new field. What's an operon? It's basically a section that controls whether or not that gene is allowed to be turned on or turned off. So not all genes will have operons, but when the ones that have them do, it's because you want to regulate whether that gene's being used or not. You can turn it on and turn it off with special sequences and special little repressor proteins, and we'll talk about that next. Down below, I'm not going to show you because I'm not going to give it away, but down below on this exact same thing, you've got this activity, and I've given you the answers down below here. So give it a try first just to see, and then check your answers and see how you did. It's not very difficult, it just takes a little bit of time. All right, moving on to the next part of your note. I actually would prefer to start with the Lac Operon, and then I'm going to come back to the Tryptophan. So we're actually going to go to the second page first. Lac Operon. Again, operon is a section of like an entire gene section. So if you look down here at the second picture, you can see that you've actually got the promoter region, then you've got something called the operator here, and then you've got the genes that you're trying to transcribe. Now keep in mind, this is a prokaryote. This is a bacteria set of genes. We don't really have our genes set like this. Okay, so keep that in mind. Here's what you need to know. When we're talking about genes being produced, so they've cut off the picture here, but it's down below. We have a promoter section, and that promoter includes the TATA box, which would be this section right here. That's where we're going to pop open the sequence. They haven't shown both double strands here. They've just shown it in like one big long loop, but there is a double strand here. You're going to pop this section open. It's rich in T's and A's. Much easier to open a T and A section, a TATA box, because there's only 2 bonds between the A's and the T's compared to 3 hydrogen bonds holding C's and G's together. So low energy to pop the section open. The very next bit is an operator. The operator is specially designed sequence that you can put a repressor on. A repressor, if it is active, is attached to the operator and shuts down this gene, because RNA polymerase has to pop open at the promoter and then begin to read the gene. So you have your little start codon, and then you're reading that gene. But if this repressor is physically in the way, this guy can't get past it. So this has to fall off first, get out of the way, leave the operator, so that the RNA polymerase can actually read the sequence and create the mRNA. Okay? So if it's attached, if the active repressor, if the repressor is active, it's another protein, it will block the expression of this gene, okay, because it's going to be attached. Well, it's a protein. It's a product produced by this particular genome. Where does it come from? Well, it comes from an mRNA. Where does that come from? It comes from another gene, a regulatory gene called the LAC1. In the LAC1, you'll find the regulatory genes are always to the left of whatever they're helping to control. So a regulatory gene is always, we say, upstream, so a little farther left of the main sequence. So you make the mRNA, then the mRNA makes a protein. Now I have an active repressor that can turn this guy off. Okay, but what's the point in having it always off? Well, the whole point of the Lac Operon, the end goal here, will be to produce enzymes that digest the sugar lactose. So these enzymes down here, so there's your codes for the DNA, here's your mRNA sequence, and then these eventually will become the 3 proteins that you're going to need. Notice that all 3 of them are enzymes, ASE ending. You need these 3 enzymes in order to, as your machinery, to help you digest lactose. Well, when would you need those enzymes? Well, you only need them when you have lactose present. If you don't have any lactose, why would you keep making enzymes that can rip up lactose that isn't there? So this gene you actually want to have always off until you need it. So the Lac Operon is always off until you need it. How do you turn it on? Well, there's another version of lactose called allolactose. Allolactose is like, allo just means other. So it's like this weird other version of lactose. It's still lactose sugar, but it's a very rare version of it. Anywhere between 1 to 5% of allolactose, is allolactose, up to 5%. So if you've got a lot of lactose, then you should have a lot more allolactose than normal, because if you have 100 pieces of lactose, about 5 of them are allolactose. So as soon as the allolactose begins to build up, that tells you there's a lot of lactose available. So then what will happen is the allolactose itself attaches to the protein repressor. So imagine that the repressor is still attached to the operator. This is its normal setup, would be attached. It's going to come and bind right here, change the shape of the active repressor and make it inactive. When it changes its shape, it falls off the operator, and now the RNA polymerase is able to run through and do its job. Okay? I'll go back down to this picture again. So imagine that this repressor is attached up here. The allolactose comes into here, it changes the shape of it, this repressor becomes inactive and falls off the operator, and now RNA polymerase is free to transcribe that sequence. Okay, that's it. That's how it works. Because we're forcing it to turn on, we would say that the Lac Operon is an inducible operon. It's forced to be turned on. So it's always off until you want it turned on, and then the thing that you want to digest is going to turn it on. It's actually pretty brilliant when you think about it. You don't want something else to be turning it on. You want the thing that you're trying to digest to turn it on. That way, when its levels go up, it turns on. As soon as the lactose levels drop, allolactose will also get digested. So that means that when this guy gets digested, then this guy here, the repressor, is free to go back and bind. And it will do that. And that will shut down the Lac Operon again. So you're always off. You're only turning on when you want to actually digest or make these end products, when you want to digest lactose. So when lactose is absent, the repressor is active and the operon is turned off. When lactose is present, the repressor is inactive. Okay, because I have induced it, this is called allolactose, the inducer, and that means the operon is turned on. The trip operon is always the example we use for the opposite idea. Whereas the Lac Operon is always off, this one is always on. So it's important to know, well, why is it always on? If you remember back to the protein section, we said that there are 20 amino acids that are required for the human body. We have 20 different amino acids that make up all of our proteins. Of those 20, 8 are essential because you can't make them. You get them in your diet. When you're a baby, you would actually say that 9 of them are essential because histidine is, you can make it, but you don't make enough to keep up with the fact that you're growing. But by the time you're an adult, there's only 8 of them that are actually essential. But that means you can make 12 of them. And here's how you make one of them, one of those 12. Tryptophan is a type of amino acid. And your end goal then is to make the machinery, so these polypeptide subunits, these guys are going to make up the machines that help me to make tryptophan. Okay? So if you need tryptophan, you're probably going to need it quite often. You're always going to want this on. The only time you don't want this on, the only time you want it turned off, is when you've got enough tryptophan, which is why it's brilliant that the end product, the whole goal of this thing is to produce machines that make tryptophan. So if there's enough tryptophan, tryptophan is going to turn it off. It's called co-repressing. So here's the basic idea. Here you've got your promoter region again, which contains your TATA box and your operator. You've got your genes that are going to be transcribed, and the RNA polymerase is free to keep making the mRNA as long as nothing is in the way here. So normally we have an inactive repressor. And notice again that the repressor is a separate protein made by a separate piece of mRNA made by a separate gene that is upstream from the main sequence. Okay, so your regulatory gene is upstream. So TRPR, okay? So when there's enough tryptophan, more than enough tryptophan, tryptophan comes in and changes the shape of the inactive repressor and makes it active. So together, they're going to repress and stop RNA polymerase from being able to make the mRNA, from being able to transcribe. That's why we call tryptophan a co-repressor. On its own, the repressor can't do anything. But if tryptophan is present, now I can stop this gene from working. Okay? So here, we've got tryptophan absent. That means I need to make more. So I'm going to continually make it, transcribe. That leaves me free to translate. Makes lots of machines that make tryptophan. Now I've got tons of tryptophan available. Tryptophan comes along, co-represses by changing the shape of the repressor to make it active, shuts down the operator, and now RNA polymerase cannot do its job. And that's how operons work. Please make sure, it's really important that you go through and you answer these questions here, and that you're able to tell the difference between the Lac Operon and the Tryptophan. There'll be lots of questions on your test coming up shortly that you'll have to be able to distinguish the difference between those. So please pay some good attention to what's happening here with the Lac Operon. Be able to explain it. Maybe even just steal somebody at home and just make them listen to you while you explain the Lac Operon and the Tryptophan. But here's your key idea here. Always off until I induce it. And here it's always on until I co-repress it. You've got to know all these new little terms. Alrighty? Have a good day.